So, Dr. Bale, both great presentations, but I was wondering, I'm sure based on what you've seen so far, you're thinking about the evolutionary disadvantages or advantages for sexual brain development in relationship to this exposure to stress and their microbiome. Can you pontificate on that a little bit for us?
How much time do we have? So I get this question a lot. Why would there be a way in which this would be transferred? Why would it be different for males and females? And I think it's a complex question. And I think if you ask any NICU nurse or any pediatrician, any OB-GYN, they will tell you there's this thing called the wimpy white boy syndrome. And that if you go to a NICU, there's a lot of more males than females. And there's something about the vulnerability of that neonatal, prenatal window that males don't seem to survive and or are more vulnerable, and females are protected. You look at autism as a good example, right? Early developmental manifestation, it's 4 to 5 to 1 boys to girls. Why is that? And I was actually at a wedding recently, and I sat next to a NICU nurse, and I said, God, I hear this all the time. Is it really true? And she said, absolutely, that the NICU would be populated with white boys, black boys, white girls, and then black girls. And there's something racially and sex specific about the vulnerable aspect of that neonatal window, which I think is fascinating. So then I asked a pediatrician who works in Philadelphia, actually in West Philly, mainly sees African-American kids in his clinic, and he works a lot with autism, and he has never seen an African-American girl with autism. And so I started putting these, so are there differences, and are they evolutionarily beneficial or not? Does it depend on the environment that you might be born into? I think there's a lot of questions there. There's a whole Barker hypothesis that the brain sets up in anticipation of said environment. And then if you don't have that environment, that's the disease risk. I think that there's factors there that are incredibly interesting, but I do think it's the mismatch of that prenatal environment, postnatal environment, and that the sex specificity probably lies along that continuum for developing rates. I showed you the differences in the immune cell populations present in the gut, where big sex differences at birth already. So yeah, that's the best I can do for pontificating it on a Monday.
Both talks were very, very interesting. I wonder, Dr. Bale, if you could expound on some of the metabolites that you said you would discuss later, maybe, and also what is it specifically about lactobacillus that might be driving sort of sex-related changes?
Those are both great questions. So many of the proteomics and metabolomics that Eldon has run at the level of the different parts of the intestine, as well as in the plasma, and then comparison to the brain, suggests that the content of the inoculant is different between males and females, and is also altered by stress, such that what they're breaking down and how they're being absorbed, which again, I will say is in large part probably related to the interaction with the host immune system, and then how they're getting transported in the brain. We haven't completed yet the studies that we're working on in terms of the blood-brain barrier, and is it shifting, again, the closing of the blood-brain barrier? Is that the difference in the transport? Right? There's whole aspects here of trying to determine, again, step by step, to make those mechanistic understanding. Lactobacillus has been shown in many different studies. So this is why Eldon has insisted that we now spend the money doing shotgun to try to get at more species level analysis of what it is. He has come to some conclusion. I'm making him repeat all that before I begin discussing it, so that we can begin to discuss what the functional, right? Because that really, and I've heard several talks on that today, which is what is the functional consequence of these changes? Because we do developmental studies, we're not just doing fecal output, we can actually look at the different parts of the intestine. So he's done a lot of that as well to ask the more functional question. So I'll get back to you on that, I guess.
So I have a question about the honeybees. So there's a set number of species, but then there's huge strain variation. And I'm wondering whether, if I understood it correctly, I'm wondering whether colonies which are in different geographic environments may be more or less fit because they have a certain complement of bacteria that are living with them. In other words, if you have a hive, is a hive better or worse off in a certain environment because it has the bacteria there that allow it to adapt properly, the whole hive, to adapt properly to the place where it's living?
Right, that's a good question. We're really very interested in that. So worldwide, you'll get the same species pretty much everywhere. Bacterial species, which just means at the 16S level, they're just a couple of percent divergent at most. But there's all these strains, and the strains do differ among hives. So like a different colonies. So to look at this, we've developed sort of high throughput markers that are protein coding. So we're not sequencing everything because you'd never get deep enough. But you can, for a given species, you can make primers that are conserved enough to, they'll be, say, 80% identical for a protein coding gene when they're 99 or 100% identical for 16S. So using these other markers, we've done some of that, and there are differences among colonies. I don't know geographically how consistent it is, but for example, the highly managed colonies versus kind of feral colonies that are not interfered with much. And I think that there's a good possibility that there is some geographic adaptation. For example, if you look at one of the species, Guillemin, it has all these pathways for using different sugars and different, depending on what plants are present in a given region that the bees use, they're going to encounter more or less of given sugars. I mean, they're very different among plants. I mean, whether they're in the nectar and so on. There's a lot of stuff. So just at that level, metabolizing food from different sources, there could be local adaptation, and those are what vary among strains.
And do these good guys that live in the bees protect the bees against pathogens?
Yeah, I didn't
Are they
talk about
part
that,
of
but there's
the-
actually quite a bit, both from us and another lab in Switzerland, that they are protective against, to some extent, they're protective against positive strand RNA viruses. And also, the most work has been done on, there's a trypanosomatid that is a significant pathogen in both bumblebees and honeybees, and they protect against that. So the mechanism, we're not sure if it's through the innate immune system. We've done a bunch of stuff on this, and it's kind of fuzzy. But there's a protective effect. We don't know how it works, whether it's a direct, to some extent, it seems to be a direct interaction between, at least in the case of the trypanosomatid and the gut bacteria. It seems to be direct and not through the host immune system, because different strains of gut bacteria will be protective against different strains of trypanosomatid. It doesn't look like the host immune system is at work. So, but there is a definitely protective effect. We've also done studies where we take some bees, give them antibiotics or controls, mark them, put them back, and treatment with antibiotics increases mortality. I mean, it disrupts the microbiota, of course, a lot, and it causes them to die more. So it suggests that, in the hive environment where there's all kinds of microorganisms, that this happens.
Thanks.
Tracy, two questions. One, do you know if the effect you see in males is hormone dependent? So if you castrate the males, would you see the same or not? Question one.
This is a big question. So we haven't castrated them because they actually have the phenotype. So they're actually dysmasculinized. So the males that are from the early prenatal stress moms that show the phenotype, everywhere there's a sex difference phenotypically in stress reactivity, whether it's the HP axis that I showed you, behaviorally, cognitive, spatial learning, and memory strategy, their phenotype is actually more in the direction of a normal female than it is of a normal male, which sort of points to, we call it dysmasculinized because they have testes, they produce testosterone, but it's like they have a little bit less testosterone and a shorter anogenital distance. So it looks like there's some aspect of not getting fully masculinized, and again in that neonatal window. So we haven't done the castration. We've discussed doing the opposite, is in terms of rescuing them by giving them back, normalizing during that window the testosterone. But there's a huge field that studies this, so I know that if we likely could masculinize it. Whether or not that really helps us out with the mechanism of what's happening, we haven't done. But I have a new graduate student that's actually now looking over the pubertal window to look at why we see these massive changes in their reduction in their growth that hits at weaning. So whether or not it's the stress of weaning in concert with puberty and maybe hormonal related is something that we're doing now.
We can do it by giving him lactobacillus that will increase testosterone level.
Testosterone.
And the second question was, does it matter that the mother during the stress has microbes or she doesn't?
So if we
If
did,
you stress the germ-free mouse,
that's a really complicated study because if for her to be germ-free, she's going to be a bit of a different creature. So we'd have to sort of recapitulate our entire phenotype and redescribe it first to figure out if that.
no, but you can take progeny from stressed germ-free mouse or not, take progeny and colonize it with stressed mouse and unstressed mouse, and it will sort of give you an answer.
So the okay, yeah. So we did the C-section study really is essentially germ-free. It's not a germ-free mom. True.
I just wanted to follow up one last question. I mean, this whole interaction between hormones and microbiome, I wonder, Nancy, if you have any comments to make about, I guess the whole determination of the queen versus the worker bees is hormonally regulated. Do you see interactions with hormones and microbiome composition in your system?
Yeah, I didn't mention it, but queens are a totally different animal, and they don't have the same microbiome at all. I mean, their whole metabolism is completely different, and they're basically egg-laying machines, and they're not food processing machines, which the workers are. And they basically don't. What the queens have actually looks a lot like the worker on its first couple of days. It's just kind of this erratic mix and has more environmental things. It's also much smaller microbiome. She's larger, but she has many fewer bacteria, and her gut is different and everything. So she's completely different for the microbiome. The males, which I also didn't mention, are sort of in between. They're the drones. They're sort of intermediate. They have a more erratic, but they do have some of the same organisms, which makes sense because they do eat and everything. So.
All right, well, I'd like to close this session and thank all the speakers for this afternoon session. Thanks.